In this video, I want to take a small tangent to discuss a fascinating result from the type of analyses I taught you about in the previous lecture. As you will learn later in the semester, many organisms cooperate to get the nutrients they need. For example, Polynella chromatophora is an amoeba that contains a tiny plastid that sure looks like a cyanobacterium. Cyanobacteria are photosynthetic bacteria that can generate food from sunlight and carbon dioxide. This plastid retains the ability to make peptidoglycan and phycobilisomes, just like cyanobacteria. In exchange for not being digested, the plastid provides nutrients to its host. The plastid is an endosymbiont, meaning that it cannot be cultured outside P. chromatophora and in fact is absolutely dependent upon it. Extracting the DNA of the plastid and sequencing it showed the genes in the plastid are very closely related to those found in free-living cyanobacteria. The genes shown here are actually genes involved in the photosynthetic process and fixing nitrogen. Again, we'll explain those processes later in the semester. The important point is that this organism is very similar to Syncococcus, which is a cyanobacterium. It looks like the amoeba ate a cyanobacterium in the recent past and formed a very close relationship. If you think about this a little bit, your next question should be, what about the chloroplast? Isn't this phenomenon the same? A plastid is an organelle inside that amoeba. The chloroplast is an organelle inside photosynthetic plants. You would be right. The chloroplast has many cyanobacteria-like properties. It has two membranes, like a gram-negative bacterium, which cyanobacteria are gram-negative. It contains a thylakoid that is the site of photosynthesis. The enzymes and proteins in the thylakoid resemble cyanobacterial proteins closely. Chloroplasts contain their own circular DNA and 70S ribosomes. These are things that are found in bacteria. They don't have 80S ribosomes as eukaryotes do. The function of the chloroplast is just like the plastid, to provide nutrients for the cell where it lives. Archaea and bacteria preceded eukarya in the lines of descent. But have you noticed that it seems as if eukarya are this curious mix of bacteria and archaea? Professor Lynn Margulis realized, though she didn't use today's terms, that an endosymbiotic relationship between two entities created eukaryotic cells. One was an archaea, the other a bacteria. The archaeal parts were the ribosomes, the membranes, the cytoplasmic machinery, and other cellular enzymes. The bacterial parts were the mitochondria and the chloroplasts. She coined the term endosymbiosis to describe her hypothesis that these organelles had bacterial origins. So what is the evidence for the endosymbiosis hypothesis? Both mitochondria and chloroplasts contain DNA that resembles the chromosomes of bacteria. Two membranes surround these organelles, reminiscent of gram-negative cell wall structure. Mitochondria and chloroplasts divide by a method that resembles binary fission. Much of the photosynthetic organelle's internal structure and biochemistry inside chloroplasts is very similar to those observed in cyanobacteria, a photosynthetic microbe. With this evidence, she proposed her theory, which other scientists initially pretty much ignored. However, over the years, more evidence accumulated. The ribosomes of mitochondria and chloroplast resemble those found in microbes. When phylogenetic analysis came along, it was possible to analyze the 16S ribosomal RNA sequence of these ribosomes. Given what we have talked about, what do you think is the phylogenetic origin of the mitochondria? How could you test this? What scientists did is they sequenced the 16S ribosomal RNA, they aligned it with that of the bacteria, and they found its place within the bacterial phylogenetic tree. They did the same thing with the chloroplasts. What they found is these organelles are in fact closely related to proteobacteria for the mitochondria and cyanobacteria, no surprise there, for the chloroplasts. This last bit of proof is convincing evidence that supports Dr. Marghella's hypothesis. What this also means is that bacteria infect every one of your cells. You would not exist if it were not for a microbial partnership that began eons ago. Here I present the most recent hypothesis of the tree of life. Unlike previous trees, this one has an even larger amount of evidence supporting the lines of descent, and clearly shows the merging of archaea and bacteria to form the eukarya. There were two such events. The first being the origin of the mitochondrion, when it melded with archaeal bacterial species to form the first eukaryote, and the second being the chloroplast, which originated in the line of plants. I like this tree because it puts each domain in what I feel is its proper place, and it also emphasizes the major role that microorganisms play in the world. With your greater understanding of phylogenetic relationships, let's now revisit each of these three domains. This slide shows an example of the tree of life for the bacteria. Again, remember that bacteria are mostly single-celled organisms. They grow by asexual reproduction. They have a simple morphology. They have complex and diverse metabolism, and a few of them are important pathogens. Let's just look at a few important groups. Firmicutes are gram-positive and contain important disease-causing species listed here. I won't read them all off. You can read them yourself. I do not expect you to remember these names now. We will cover pathogens at the end of the semester. Some are endospore formers, and many are important in food production and biotechnology. Lactic acid bacteria are important for fermentation of many foods, including cheese, sauerkrauts, pickles, kimchi, kefir, kombucha, and many other types of fermentation. Images at the left show some images of the lactobacillus species. The proteobacteria have a gram-negative cell wall and also contain many disease-causing bacteria. This is a very large group of organisms. It is from this phylogenetic group that mitochondrion originated. Several of these species are important for biotechnology. The actinobacteria have a gram-positive cell wall and grow by forming filaments, or long tubes. They do contain a few disease-causing members, most notably species that cause tuberculosis. However, this group is also the source of many useful antibiotics. The cyanobacteria have a gram-negative cell wall. They often grow in colonial or multicellular groups. While none are known to cause disease, they can produce toxins that can lead to deaths of animals and humans. They originated oxygenic photosynthesis, meaning a metabolism that produces oxygen, which led to a complete change in our atmosphere. Cyanobacteria are related to chloroplasts. They are also called blue-green algae. The archaea organized into two main groups, Eurarchaeota and Crenarchaeota. Again, the archaea are mostly single-celled organisms, grow by asexual reproduction, have a simple morphology, have a complex and diverse metabolism. None are known to be pathogens. The original archaea isolates were from extreme environments, hot springs, high salt environments, or high acid environments. Further research has found them to not be limited to these places and actually be found in many different environments. They are present in vast numbers in the soil and in oceans. The left image shows a core of a methane oxidizing archaea, stained red, that uses methane as an electron acceptor. The right shows a termite that has methanogens, the little yellow organisms in panel C, that take acetate produced during digestion and convert it to methane. Methanogens are one of the important groups in the archaea. They convert hydrogen and carbon dioxide to methane and release it into the environment. They cannot survive in the presence of oxygen and are present in many different environments. Several examples of methanogens are shown on the left, while the image on the right shows scientists connecting swamp gas, methane, and igniting it. Who said science is boring? Eukarya are the third branch of the tree of life, but even here, Most of the life is microbial. Only animals, fungi, and plants form macroorganisms, a small part of the tree of life. There are all sorts of interesting protozoa, and many of the lineages have not been worked out completely. One thing to note is that the grab bag term protist does not refer to a coherent lineage. Remember that eukarya have a membrane-bound nucleus. Some are single-celled organisms, some are colonial or multicellular. They reproduce by asexual or sexual reproduction. They have a simple or complex morphology depending on the species, and they have a relatively simple metabolism. Let's look at a few groups. Many protists are microbial. Most of them are harmless to humans, but a few cause important diseases including Giardia, Leishmania, and Trypanosoma. Another important group of eukaryotes are the fungi. These are absorptive heterotrophs. Most are saprobes that recycle organic material. Often they live dual lifestyles, one unicellular and a second as a multicellular organism. The second macroorganism is often when they reproduce via spores. There are a few that cause disease, and they are especially important for plant diseases. Fungi are also important for biotechnology and food production. Some fungi are dimorphic, living a double life. One in a yeast morphology that is unicellular, forming oval-shaped cells. The other is filamentous, that forms long tubes. There are many famous fungi. Saccharomyces cerevisiae is important in beer and bread formation. Molds and mildews spoil lots of produce and other things. Mushrooms can be edible or poisonous. If you do pick wild mushrooms, make sure you know the difference. Before we end this lecture, I would like to bring up one more idea. First, I want you to think of the conventional definition of a species that you learned in your biology class. Please write it down. Most often, students will think back to their biology class and come up with a definition that has something to do with members of a species being able to interbreed. That definition will not work for prokaryotes since they only reproduce asexually. So how do we define a species? First, scientists attempted to do this by comparing what they could do or how they looked. In other words, by their phenotypes. You can bet, just like in classification, this didn't work too well. Organisms that have similar properties were thought to be a species. The more traits you have in common, the closer your relationship. This way of organizing doesn't work that well because bacteria can lose and gain abilities, yet still be very similar. Next, they tried examining the DNA. Before DNA sequencing, they would run hybridization experiments. The higher rates of hybridization between the chromosomes, the more related to organisms were. An arbitrary cutoff of 70% hybridization was used to define a species. With the advent of DNA sequencing, it became possible to compare 16S ribosomal RNAs and then eventually even more of the organisms. Again, an arbitrary cutoff of a 97 to 99% similarity for 16S ribosomal RNA denoted a species. However, based upon this definition, all primates are the same species. Trying to determine a species in bacteria gets even more complicated by extreme genetic diversity in microbes. Even organisms sharing identical ribosomal RNA may have different genes and therefore different properties. How can this be? It turns out bacteria will happily exchange DNA through various mechanisms. We will talk about how later in the semester. They will sometimes then incorporate this DNA into their genome. This phenomenon is called horizontal gene transfer and is in contrast to vertical gene transfer, which is the set of genes you obtained from your parents or parent cell. As you can imagine, this will significantly complicate figuring out lines of descent. In the past 2 decades, research has sequenced the genomes of thousands of bacteria. One example is Salmonella, a bacterium that causes a nasty gastroenteritis. Look at the substantial differences in the number of coding regions within these species. Again, this is the same species in a lot of cases, right? They give them different names, but they're all Salmonella enterica. The variation is all within one species. There are numerous events of horizontal gene transfer within this species, and it is the same for most organisms. This organism seems to have a core genome of 4,316 genes, but then other members of the species have all sorts of other genes. What are the implications of horizontal gene transfer for evolution? Remember the classic pathway of evolution. Each species has natural variation due to mutations each individual holds. Natural selection then acts with advantageous mutations winning and deleterious mutations losing. Horizontal gene transfer messes this up. Now, at any time, a bacteria may acquire a set of genes that suddenly make it fitter than other members of the species. For example, the ability to degrade an available carbon source. That organism would then be dominant over all the organisms that couldn't degrade it. Horizontal gene transfer appears to speed the process of evolution, and horizontal gene transfer is one reason that drug-resistant strains of bacteria to antibiotics appear so quickly. Before this realization, we had a nice pretty picture shown at left where lines of descent followed orderly patterns producing the organisms present today. Horizontal gene transfer is an irritating phenomenon that messes this all up. The real tree of life is probably more like the image on the right. The question is, how much does horizontal gene transfer alter the tree of life? We are still trying to sort out what this means for the tree of life, and also what does it mean to be a bacterial species. This brings us to the end of the lectures on phylogeny.